Hello, Smart Medicoz. Today we're doing a deep dive into, well, one of your body's really sophisticated defense teams, the complement system. Think of it like your immune system's special ops crew and alarm system combined. It's this complex group, over 20 plasma proteins, C1 to C9, and others, always on guard.
Yeah, and what's really striking is just how versatile it is. It's critical in both the immediate innate response and the more tailored adaptive immunity. And it works in this really interesting cascade, like dominoes falling. One protein activates the next, leading to these big outcomes, inflammation, helping cells eat invaders, even directly popping pathogens.
Right, and you mentioned a core step, something about C3.
Exactly. The absolute heart of it is C3 cleavage. When C3 splits into C3a and C3b, that's what really unlocks its power.
Okay, so how does this whole thing get triggered? It sounds complex.
Well, there are essentially three main ways it gets switched on, different triggers. First, you've got the classical pathway. This one needs antibodies. It starts when C1 binds to IgM or IgG that's already latched onto an antigen, an invader.
So antibodies flag the target, and C1 sees the flag.
Precisely. Then there's the alternative pathway. This one's more direct. It gets activated straight by microbial surfaces, things like LPS on bacteria. No antibodies needed initially.
Okay, skips the middleman.
You could say that. And finally, the lectin pathway. This kicks off when a protein called MBL, mannose binding lectin, recognizes specific sugar patterns on microbes.
So 3 different reconnaissance methods, essentially. But they all lead somewhere specific.
Exactly right. They all converge, funneling down to create something called a C3 convertase. That's the enzyme that does the crucial C3 split we talked about.
Yeah.
C3a and C3b.
Got it.
And C3b doesn't stop there. It helps form another enzyme complex, the C5 convertase. This then splits C5 into C5a and C5b. Again, 2 important pieces. C5b starts building the MA, the membrane attack complex.
That sounds destructive.
It is. That's the part that drills holes in microbes. C5a, on the other hand, is a major player in inflammation. It's like a potent distress signal. The MAC itself, C5b, joining up with C6, C7, C8, and multiple C9s, embeds right into the microbial membrane, forms a pore, water rushes in, and bang, osmotic lysis. The cell pops.
Wow, okay, direct destruction. But you mentioned C5a and inflammation and C3b doing other things. It's not just about lysis then.
Oh, definitely not. Lysis is huge, but complement does more. Inflammation is a major one. C3a and C5a are called anaphylatoxins. They trigger mast cells to release histamine, make blood vessels leaky so immune cells can get in easier. And C5a specifically is a powerful chemoattractant. It calls neutrophils and monocytes right to the battle scene.
Like a flare signal for reinforcements.
Exactly. Then there's opsonization and phagocytosis. This is where C3d really shines. It and a breakdown product called IC3b coat the microbe. This coating acts like eat me signals for phagocytes, macrophages, neutrophils. It dramatically enhances their ability to grab and engulf the pathogen, makes cleanup much more efficient.
So tagging for destruction and signaling for help. And the direct kill with MA. That covers a lot of ground. But, with something that powerful, how does the body stop it from attacking our own cells? There must be controls.
Absolutely critical question. You can't have the system running wild. There are several key regulatory proteins, for instance, C1-INH or C1 inhibitor. It blocks the classical pathway right at the start. If you lack this, you get hereditary angioedema. Then you have proteins on our own cell surfaces like DA, decay accelerating factor, and CD59. DA disrupts the C3 convertase, and CD59 blocks the MACE formation. If you lose these, like in paroxysmal nocturnal hemoglobinuria, PNH, your own red blood cells become vulnerable to complement attack.
So protective shields on our own cells.
Right. And there's also factor H, which is a major regulator of the alternative pathway in the fluid phase and on cell surfaces. Mutations here, they're linked to serious kidney issues like atypical HUS and also macular degeneration in the eye.
It's such a tight balancing act then. Which brings us to, well, the clinical side. What happens when this balance is lost?
Well, if you have complement deficiencies, you're often more prone to infections. Deficiencies in the MA KIC components, C5 to C9, famously increase susceptibility to Neisseria infections like meningitis. And deficiencies earlier in the cascade, C1, C2, C4, they're actually strongly linked to autoimmune diseases, particularly lupus.
Interesting. So too little can cause problems, but what about too much?
Right. Overactivation is also a major issue. It can drive autoimmune diseases where antibodies mistakenly trigger complement against our own tissues. And as we mentioned, conditions like PNH result from losing the regulators, leading to uncontrolled complement activity. But the flip side is, because it's so central, the complement system is now a really hot therapeutic target. Drugs are being developed to inhibit specific parts of the cascade for autoimmune disorders, kidney diseases, blood disorders. It's a major focus in medicine now. So just to wrap that up, all pathways funnel to C3. Cleavage gives you inflammation signals, C3a, C5a, pathogen tagging or opsonization, C3b, and the cell-killing MAC, C5b9. It's powerful, essential, but tightly controlled. And when that control fails, either way, you see significant diseases, autoimmunity, infections, hemolysis.
It really highlights the immune system's incredible challenge, doesn't it? Constantly having to tell friend from foe with such potent tools. So thinking about that fine balance, how might a deeper understanding of this system, its checks and balances, lead to even more precise medical interventions down the line? Something for you all to ponder.